Assalamu alaikum wa rahmatullahi wa barakatuh. Today we will continue with the lab 4, and this week we have third week of embryonic development. So as you know that we've finished second week of embryonic development, and we've mentioned about some events that happen in the second week of embryonic development. So this week we will have other group of events that happen or what will form in this week, I mean the third week, but we will take it as some definitions or events, and you're gonna study it in detail in the theoretical embryology lectures. So first of all, you have a term that we call it gastrulation, and gastrulation is actually a process. Okay, it is a process. Even gastrulation هي عملية that will establish all three germ layers, ectoderm, mesoderm, and endoderm, in the embryo. So you will wonder, what does it mean, ectoderm, mesoderm, and endoderm, and what are the germ layers؟ يعني شنو هي هاي الجيرم layers؟ هي احنا في الحقيقة عندنا ثلاث لايرات أساسية اللي هي ectoderm, meso, endo، ثلاث طبقات اللي راح ناخذها in details، ونشوف انه هذه الطبقات نفسها هي راح تكون all tissues and organs inside the embryo body. إذن بصورة عامة هذا الأسبوع يتميز بعملية اسمها gastrulation. So generally this week is has a characteristic, an important characteristic, which is include gastrulation. Okay, so gastrulation is actually a process that will give us or establish or form three germ layers that are called ectoderm, mesoderm, and endoderm. Okay, so let's come to the structure of the embryo itself. And we stopped last time and we said that we have epiblast and hyperblast. It was as a shape of the ball, spherical, isn't it؟ كان عندنا فد structure هو كان كروي، صح لو لا؟ كان عندنا من الجهتين خلينا نقول cavities. Okay, from this side and this side إذا تذكروها، من cavity and exocelomic cavity. فاحنا صراحة شو راح نسوي هسه حاليا؟ تخيلها انه نقص هذا الكرة من المتصل من الجهتين، خلينا نقول الكافيتيات نخليها على حدة، وناخذ اللايرين هذه hyperblast and epiblast. Okay, so just mention that imagine that you have just these two layers without the cavities, and we will just look what happened in these layers, or what are the changes that may happen in third week of development. So as you see, the blue one is epiblast, the blue surface, and the yellow surface is the hyperblast. Okay, so look at here, in the blue surface structures will form, and these structures are so important. Why? Because later on we will see that all parts, other parts, I mean the parts that will form the organs, the origin for the organs, will form from these structures. Okay, so first of all we have a structure, we call it primitive streak. Okay, streak means a line. Okay, so this is the primitive streak, and this is the slightly swollen region. Okay, on each side of the primitive groove, and formed by aggregation of epiblast cells. So as we said that this surface is the epiblast cells. Okay, هذا سطح تحتوي على epiblast cells. So these epiblast cells will usually aggregate, تتجمع, and they will form a groove. Okay, a groove شنو هو؟ كأنه خندق يكونون, and they will form a streak, and we call this streak as primitive streak. Okay, so let's continue. Second thing, we will have a primitive node. A primitive node is a slightly elevated area formed by collection of epiblast cells in the cephalic end of the primitive streak, and acts as organizer. So look at here, as it say that it's an elevated area، منطقة شوية تكون مرتفعة عن surface itself, and they will form by collection of epiblast cells. مثل ما تشوفون مع انه هو على السطح، سطح الابيبلاست، فهو يتكون من epiblast cells. يعني ممكن اسألكم what's the origin of the primitive node, and you're gonna answer it's the epiblast cells. Okay. So this is in the cephalic end of the primitive streaks, and it has a function that is organizer. This will organize many processes that will happen later on. Okay. And you have a primitive pit. Okay, the primitive pit, it will be inside, okay, inside, and it's depressed area inside this primitive node. Okay, a depressed area inside the primitive node. Okay, so let's come to the gastrulation process. Simply gastrulation process, it is it includes two processes, and they are so important to know. That is one of it migration, and another one is invagination. So أول شيء عندنا migration اللي هي هجرة الخلايا. Okay, then invagination. Okay, then invagination means they will detach from the surface and come on, slip beneath it, inward the movement. Okay, so the cells of epiblast cells, I mean, that is found in the surface, they will migrate toward the primitive streak. So look at here, this is the primitive streak, and they will all migrate toward this part. Okay, I mean the primitive streak. So this movement of the epiblast cells from the surface to the primitive streak, we call it migration. Next, we will have invagination. So invagination, these migrated cells that are found in the primitive streak, they will become flask shape. Okay, and they will detach from the epiblast. كلهم راح يعني شو نقول يعرفون الابيبلاست، أو إنه يتركون المكان اللي موجود به الابيبلاست and they will slip beneath it through inward movement. يعني كلها كأنه تتزحلق، خلينا نقول هيك بهالمصطلح حتى يبقى ببالكم، تتزحلق بالضبط الطبقة اللي تحت الابيبلاست. تمام، تبدي انه تزحلق او انه تغير مكانها، وشون خلينا نقول انه تترك الطبقة اللي فوق اللي هي ابيبلاستس. يعني تجمع الخلايا، هجرتها وتجمعها الى primitive streak، ومن ثم انها تترك تيجي المنطقة وتروح تحت هذه شو اسمه، خلينا نقول السيرفيس. Okay, then تعريفين you have a migration and invagination. They are both so important in the gastrulation process. Okay, so if I ask you dash and dash are important steps in the gastrulation process. Okay, زين خلينا نجي نشوف هاي ثلاث لايرات كيف تتكون، اللي هي ectoderm, mesoderm, endoderm. Okay, mesoderm, ectoderm, mesoderm and endoderm. طبعا احنا هذه من ايش تتكون؟ هسه راح نشوف من اي طبقة تتكون، من اللي تتكونها، وليش تتكون. ليش؟ لانه احنا عندنا حالة تطور، and we have a development processing. So this development will have some cells or some layers of cells that will be origin for the other tissues. So the division, the formation is continuing during the weeks of the development in the embryo. Okay, so some epiblast cells will displace the hyperblast cells and create an embryonic endoderm. So first of all, we need to know how endoderm will form. Okay, how endoderm will form. So some epiblast cells displace the hyperblast. Okay, let's see right here. So this is the hyperblast cells, isn't it? The yellow one. هذه hyperblast layer وهذه epiblast. فبعض الخلايا راح تجي من وين؟ راح تجينا من epiblast. فشو تسوي؟ راح تزيح الهايبوبلاست، تقولي الهايبوبلاست وخر، اعطيني مجال اريد اني اكون بمكانك. اوكي، فش راح تسوي؟ هذه الابيبلاست سيلز راح تجي تاخذ مكان وتكون طبقة خاصة اوكي بيها، اللي هي راح تكون بعدين مستقبلا ابيبلاست اندوديرم. اوكي، سو اذا شو راح نقول ان بعض الخلايا من ابيبلاست سيلز displaced تزيح الهايبوبلاست، تزيح الهايبوبلاست سيلز and create the endoderm. إذا أسألك شنو هو origin، ما هو منشأ endoderm، راح تقول لي epiblast cells. وليس hyperblast، لأن hyperblast cells مجرد خلايا مزاحة. Okay, so cells lie between the epiblast and newly created endoderm form mesoderm. Okay, إذا مجموعة ثانية من الخلايا اللي راح تبقى فوق الاندوديرم اللي هي أساسا طبعا من الابيبلاست راح تكون لنا ميزوديرم. زين السيل remaining in the epiblast والخلايا اللي راح تبقى بابيبلاست، طبعا هي ابيبلاست ما راح تخلص اللي بالفوق، الخلايا اللي راح تبقى بابيبلاست راح تكون لنا ectoderm. So the cells remaining in the epiblast, they will form ectoderm. So these cells will give rise to all the tissues and organs in the embryo. تمام. هسه شفنا شلون انه من الابيبلاست تكون عندنا اندو ميزو اكتوديرم. اوكي، بعدين راح نشوف انه اندوديرم راح يكون لنا مجموعه من organs and structures in the body والخلايا والانسجه. راح نشوف ميزوديرم راح تتخصص بتكوين special tissues and ectoderm also. اوكي، زين بقالنا انه نعرف انه بهذا الاسبوع راح يتكون لنا two important or cavities. تمام، واحدة من عندهم in the upper part and the other one is in the lower part. Okay, so one of it is oropharyngeal membrane. So what is oropharyngeal membrane? Is a small region, okay, in the cranial end. طبعا هذا احنا نسميه cranial end وهذا احنا نسميه cephalic end for the embryo. إذا the cranial end is small region of ectoderm and endoderm. إذا هذا الجزء يحتوي على الectoderm و endoderm cells, okay, at the cranial end of the disc, and they will in the future form oral cavity. تمام. They will form in the future the opening of the oral cavity. تمام. إذا سألتكم what's the origin of the oral cavity, you can answer is ectoderm and endoderm. Okay. Okay, so the next one is CLocal membrane, and this is also formed by the tightly adherent of the ectoderm and endoderm, and they will form their location is in the caudal end موجودة في نهاية خلينا نقول نهاية الامبريو, and they will form the anus opening in the future. يعني فتحة المخرج راح تتكون في هذا الجزء من الامبريو. إذا نحن نسميه CLocal membrane. كل هذه راح تتكون في الأسبوع الثالث إن شاء الله. الأسبوع القادم راح نكمل lab 5 and we will see what will happen with these embryos. Okay, with the embryo and what is the change and new events. Okay, so see if you have any question, please feel free to ask anytime. See you next week.